Good evening, everyone. Thank you for joining us tonight for the next event in our RiverWatch Winter Speaker Series. For those of you who might be new to the Illinois RiverWatch Network, RiverWatch is a citizen science organization that trains volunteers to monitor water quality in their own communities. The mission of RiverWatch is to safeguard the future of Illinois rivers and streams through stewardship, education, and sound science. If you're interested in becoming a citizen scientist yourself, please consider coming to one of our spring workshops that will be held in person across Illinois in the coming months. Registration is now open and workshops are starting to fill up, so sign up soon to reserve your spot. Now, before we get started, I'm going to ask you all to keep your microphones muted during the presentation to limit any background noise and distractions. If you have a question along the way, you can either put it in the chat box and then Sarah can answer that at the end, or you can save your question and ask it aloud during the Q&A session. And so with that, I would like to welcome Sarah Douglas. Sarah earned her bachelor's degree in biology from Hanover College in Indiana and a master's degree in environmental science from Taylor University, also in Indiana. Sarah joined the Illinois Natural History Survey as a mussel field biologist in 2008, and she currently works for the Illinois Natural History Survey as an associate malacologist in the Urban Biotic Assessment Program, where she conducts mussel surveys primarily across northern Illinois. She also conducts research on the conservation of freshwater mussels and riverine ecosystems, including work on invasive mollusk species. And so I would like to thank you all for joining us this evening, Sarah, and we look forward to learning now about freshwater mussels.
Thank you for the welcome. I'm glad to be here. Thank you everyone for joining the talk this evening, and appreciate your interest in freshwater mussels. So I am going to provide a brief natural history on freshwater mussels, their importance historically as well as currently, and then provide some highlights of some of the mollusk-related projects that INHS has been a part of in Illinois. So firstly, what is a freshwater mussel? Technically, it is a bivalved, so it's two valves, mollusk that lives in fresh water. They have a marine counterpart that we're probably very familiar with. That's also called a mussel, or sometimes clams. Our freshwater species can also be referred to as freshwater clams, naiads, unionids as a general term. It's a scientific name for the group that most of the mussels I'm talking about today belong to, the Unionidae. So one of the most unique aspects of a mussel's natural history is their life cycle. Freshwater mussels, most that is, need a fish to complete their life cycle. Here, larval mussels called Glaucidia, on the right-hand side of the illustration, are released into a potential host fish. Glaucidia usually attach to gills, but they can also attach to fins. The larval mussels will then encapsulate and hang out on their host for a few weeks while completing their metamorphosis. Once completed and now considered juveniles, they slough off their hosts to settle into the substrate. As we can see here on the left-hand side of the illustration, where we have a young mussel that settles to the bottom and then continues to grow and become adult mussels, hopefully. So some mussels will use a suite of species, say for example, black basses, and then other species will only use one or two hosts. Like in Illinois, we have several mussel species that will only host on the freshwater drum. One interesting mussel only hosts on the common mud puppy, and it's called the salamander mussel. It's the only known mussel to host on an amphibian. There are several species who can use different fishes as hosts, and these are considered host generalists. And these are like a wide variety of hosts, different fish species, I mean. Those mussels are considered as host generalists. So on the next couple of slides, we're going to look at some life history strategies on how mussels may attract their hosts. So in this video here, we're looking at a female lampsalis, and she's displaying a fish lure. You can let me know what you think this fish lure mimics. What kind of fish do we see here? And in this video, Velosa iris, which is the rainbow, both we have Lampsalis and Velosa iris species in Illinois, is mimicking, oh, there we go, a crayfish. Now you can see how this mussel is also actively moving. Let's play that again. And mimicking this crayfish in order to attract their hosts. So here on the left-hand side, we'll start are some various mantle displays. And this is in a group of mussels in the tribe Quadrillini. So a lot of these mussels are more of the ridgy and knobby pustulose mussels. On the left-hand side in A, we start with rabbit's foot. And here we have a little bit more of a diminutive mantle display. We move to B, which is pimpleback, and C, which is a winged maple leaf. We have a maple leaf that's common in Illinois. And then D is the pistol grip. Now, B, C, and D are species that will all host on catfishes. Now, in A, Theloderma species, the rabbit's foot here and monkey face that we have present in Illinois will host on cyprinids. So one of the interesting research that's found for some of these quadriline species, especially the ones that host on catfish, is that they also will have olfactory cues to help attract their host. Now, other ways that they help attract their host are by these really, really unique conglutinants. And conglutinants are simply egg packets that hold mature Glaucidia, in this case, mature larvae. And on the top left is an A. Here we have an example of Lampsalis cardium. And so this is a similar Lampsalis species to the lure video that we watched earlier. And so, you know, what they can do is either have these packets that they can eject into the fish when the fish strikes the lure, or sometimes mussels will free release these Glaucidia, these conglutinants. And we have some examples of that in B, which is the Wabash pigtoe, a close-up of an example of what their conglutinants look like. C is conglutinants from the Western fan shell. The Western fan shell isn't present here, but we have historically had fan shell in some of the large rivers. And D is an example of fluted kidney shell, which we have a different Tychobranchus species here. Its common name is just kidney shell. But you can see here how they mimic larvae that, you know, fish would like to go for. So here's some placement for where Glaucidia will hang out on the fish fin on the upper left-hand corner. And then here's some examples of Glaucidia that's been encapsulated in the bottom left-hand corner. And then on the right-hand side, we see Glaucidia being brooded in specialized chambers of the gill called marsupia. And this species is the three-horned wartyback, which is common in Illinois. And all these little white pinpoints are Glaucidia. So it's most mussels or the females, they'll have different chambers. Sometimes it's only certain portions of the gill. Sometimes it's the entire gill, depending on the species. And then we end up with these beautiful little babies. Here is an example of 11 species of young mussels that were encountered during one of our surveys here. See if you can name them. So these are very young mussels. They could probably be considered subadults. Most of them were probably only a year or two, maybe less, maybe a little bit older on a couple of them. But you know, real quickly, one of the things that we, especially when mussels are younger, we look at their growth rings. Some of these darker lines that are laid down. Some of them are really distinct and some of them aren't. So here's, it's always fun trying to find these babies. Now, Another interesting aspect of their natural history is that they are filter feeders. So what does this mean? Well, mussels filter water through their bodies to feed and breathe. And they filter out suspended particles in the water. So we can think of them as livers of the rivers. This is how our livers process toxins and chemicals in our body. So here in the middle, you can see an example of the mussel feeding cycle where algae and bacteria and sediment go in. They can also filter detritus. They have phytoplankton, zooplankton, other microorganisms. And they take what they want from that as food sources, and it can be some combination thereof, and then out comes clean water. Now, how they do this is on the right-hand side, you could see the mussel apertures. They have an in current and an X current. Sometimes you'll see the term siphon being used. So there's growing interest in using the natural filtering capacity of mussels to pre-treat water for human use. For example, a study by Teresa Newton and her group in 2011 had estimated that mussels in a 480-kilometer reach of the Mississippi River, this is part of the upper Mississippi River, can filter approximately 53 million cubic meters per day, while a Minneapolis-St. Paul wastewater treatment plant produces wastewater flows at 0.7 million cubic meters per day. And this interest extends to using mussels or other freshwater bivalves to selectively remove disease organisms, contaminants, for example, from water supplies. And it's a rapidly growing area of inquiry. And then in another study, Ismail et al. In 2014 found that the California floater is an example of a species on the West Coast, and Corbicula fluminea, one of our invasive species that's everywhere in the United States, can remove pharmaceuticals, personal care products, herbicides, flame retardants, et cetera, like that from the water. And they'll either biodeposit it in their pseudofeces that they will excrete, or that they will store them in their tissue. So let's move on to touch on some importance of freshwater mussels. So for prehistoric use, humans have harvested freshwater mussels in North America as early as 10,000 years ago. Prehistoric peoples ate the soft tissues and they used the shells to make a variety of tools and ornaments. Here's some examples of some of the shell finds in Illinois. And then in another example, in the Pacific Northwest, mussels were harvested extensively for food by Native Americans well into the 20th century. And I'm going to point you to a lot of great information on the next, this slide and a couple more slides. The Illinois State Museum has some great information on prehistoric use and button industry, especially. So next we have the pearl button industry from the late 1800s to about 1945. It became very prominent in Illinois. The Illinois River was considered one of the most productive mussel streams per mile in the US. And hundreds of local Rivertown residents then became part of this mussel industry. They fished for mussels and cooked. And then there's lots of unique tools. You can see the braille here on the lower left-hand side of a mussel harvester in Pearl, Illinois. And this photo comes from Marshall County Historical Society, from the Illinois State Museum Archive Collection. And you can see up in the upper left, the big pile of shell of mussels that's been processed from the river. So also prior to the button industry, there was a big pearl rush too. And then once the part of the pearl rush had decreased and realizing all this shell left over, that's when this invention with buttons, being able to use mussel shell as buttons, kind of came about. And so, for example, on the lower middle is ebony shell after button blanks were cut. There's different size blanks were measured, considered as lines, and these were then used for either like adult clothing or children's clothing. On the right-hand side is a photo of size 16 line buttons, and these would be for children's clothing. And again, if you check out the Illinois State Museum's website, they have a great online exhibit right now with Harvesting the River. I'd encourage you to go check that out. Lots of really cool photos, archival information. And so next we have the cultured pearl industry from 1960s until present. Freshwater mussel shells were used to seed pearl oysters. These pieces of the freshwater shell were called nuclei. And then the oyster would secrete a new layer of nacre around that mussel shell nucleus. And then it would form that cultured pearl. So the freshwater mussel nuclei were shipped to Japan, which was where the main demand had occurred for cultured pearl production. And then we have actual freshwater cultured pearls. And the first experimental US freshwater cultured pearl farm was in Tennessee. And here, the Tennessee River Freshwater Pearl Farm. And it was developed by and established by John Leighton Dress in 1963. So these pictures are from that farm here in the top left. These are the three main commercial species that are still considered commercial today. The three ridge on the very far left, ebony shell, and washboard, excuse me. And then up here are the size rings for regulation. So if they fit through there, then they have to be put back. And on the bottom left is an example of the cultured pearl. And that one is in a washboard. And then on the upper right photo is an example of the different styles and shapes of a freshwater cultured pearl that this farm has produced. Ecosystem services, what does that mean? Ecosystem services are the benefits that humans derive from ecosystems. So starting on the upper left, biomass. A freshwater mussel biomass can exceed all other benthos by an order of magnitude. In the dense mussel beds, if we have a really dense mussel bed in places, and sometimes we'll see this more in larger rivers, they can provide substrate stability in the river. There's greater effects on nutrient cycling, for example. The more dense, the more abundant mussels are in a bed. There are natural biological filters, which we've discussed previously, a little bit of their filtering impact, but additionally their biodeposition of filtered materials and dissolved nutrients can increase food availability for other organisms. And their dead shells can provide spawning habitat and shelter for many aquatic organisms. So in the photos for the spawning habitat, we can see here there's a bunch of fish eggs that's been laid on this white heel splitter, I think. And then also another white heel splitter or three ridge hinged shell that has a little, I think either a stone cat or a little mad tom that's hanging out right there. And it's kind of funny how often we'll come across that. Next, freshwater mussels are bioindicators. You can see all the yucky black gunk right in there. So, you know, they serve as sentinels in water quality. So if there's a mussel killer and similarly with a fish kill, we know something major has happened. Next is economic significance. So this is just a snip of an article that I had found online. I knew of this research happening a little bit, but the city of Philadelphia and 5 other organizations committed to create a large-scale mussel hatchery to bring millions of baby mussels back into the Delaware River. So the premise of this is that by using these living organisms, the Philadelphia Water Department is adding what they call blue infrastructure to help them do this work of cleaning the water. Philadelphia gets part of its drinking water from the Delaware River. And so we know that the mussels can filter 10 to 20 gallons of river water a day, as it was explained here. So thinking about, okay, well, if a few can do this, how much can millions of mussels filter? And so they're trying to think of this in the sense of like many wastewater treatment plants, pre-treating water before it gets pulled into water treatment before human use. So it's fascinating. It's really exciting. It's really interesting to think about all these agencies and research that's being intersected to be able to provide this. So I'm excited to see where that goes. Mussels are also a food resource for muskrats and otters and raccoons, for example. And then they provide habitat and some other effects on benthic density and community structure. So it's really interesting. You know, I don't know what species of leech this is, and I couldn't remember, but there's some interesting research and like leech communities on freshwater mussels. And almost every time I've found a big leech like this, and it ends up either I pull it off or when we process or it drops off or whatever, we find babies underneath it. So it's really interesting to think about that. Okay, so let's highlight their importance in terms of conservation value as well. Freshwater mussels occur worldwide, except for Antarctica. Unionidae, the main group of mussels that I'm talking about today, are, which is in blue here, the most cosmopolitan family in the order of freshwater mussels. And there's approximately over 800 currently recognized species. And then I actually just, I think just this week, and I hadn't had a chance to look at it, but Graff and Cummings had updated this paper that had come out. So if you're interested in that, you can get a hold of me and I can track that paper down. So moving on to a little bit more of local diversity. So in this example here, we're looking at species per region. And so the darkest green is the least speciose region, whereas the red is the most speciose. And so we have in North America, you know, our hotspot, our biodiverse hotspot for freshwater mussels. And this is important to remember for the information on this slide. This is a recent, well, I guess it was early 2000 status of biodiversity that's in the United States. And right at the top here is freshwater mussels. And if you take a look at the other species up here, almost all of the, well, all of these are either semi-aquatic or aquatic, as far as being some of the most imperiled taxa groups that we have here in the United States. Okay, so I'm going to switch a little bit of gears and talk about some of the mollusk projects that we've had. So when I started at the survey, I had worked on a statewide basin survey with my colleagues Allison Stodola and Diane Shastine on a state wildlife grant. And this was the first statewide basin surveys across the state. And we were investigating mussel communities in Illinois' wadable streams, and we were updating distribution records. And so we also did some survey work, a little bit more like targeted survey effort in 2013 as well. But we hit over 1,000 sites and a lot of person hours, every basin in Illinois. And so on the right-hand side is you can find these basin reports on our website. And you can find these distribution maps on our website as well. So if you're interested in what's near you, take a look at that, see if you can find that, download it, and that'll give you a pretty good idea of what mussels are present or had been present in your area. And so from that work and then other survey work that my colleagues Kevin Cummings and Jeremy Tiemann also do, we have updated distributions and some new occurrences. So in some of our basin survey work, we discovered that Lampsilus hydaena is present in Illinois. So on the left-hand side here, that's Louisiana fat mucket. And on the right-hand side is Lampsilus silicoidea, and that's the common name is simply fat mucket. And so you can see how similar they look. And And so we just have this cryptic species here it's probably been here for a long time, and it's just been overlooked. There's some places where both of these species co-occur. And then a couple of basins, we've determined that it's actually Louisiana fat mucket that's present, and not fat mucket, which was what had been thought before. So the known range is here in this yellowish orange here in the south. And so the lower part of southern Illinois is where we discovered this new occurrence. And speaking of new occurrence, Kevin and Jeremy had discovered bank climber in the Ohio River, just right down here in 2012. Yes, check my notes. Yes, in 2012. And so that was an exciting find. But in this case, this was just a documented range expansion. Bank climber is also pretty common in the South, and we have seen in other research, I believe, that there's been some expansion in its distribution as well. And then a new occurrence that we discovered in our basin surveys was a southern population of little spectacle case. And again, this was an example of there just really hadn't been that many surveys done in southern Illinois. They're hard basins to survey, for one, hard rivers to survey sometimes. And then there's snakes that live in some of those rivers that previous naturalists hadn't really wanted to, I'm talking about like in the 50s or earlier 70s, hadn't really wanted to do much. And there hadn't been a concentrated statewide effort before, but there's been lots of different sporadic surveys for freshwater mussels across the state. It's incredible the amount of information that we've had. So again, here it's just a lack of information, a lack of surveys, that these species have probably always been in this part of the state. So that was exciting to expand that range. This information actually helped and to inform these little spectacle case had been a listed species, but given the information that we found and the population numbers that we found in the basins, they were just delisted last year, actually. So one of the other fun things that we can do on occasion are in this example here on the upper left is when there was some major flooding on the Illinois River in, what was that, 2013, early 2013, and those barges had broke loose. They had got stuck up against the Marcel's Dam. And so a little bit later that spring, they were lowering the river in order to get the barges out and then check the dam for damage. So the Army Corps of Engineers had called us, and in an effort also with IDNR and Fish and Wildlife staff to be able to come and survey part of the river just to see what was there. We really haven't had too many big river surveys. William Sterrett in the 70s had done an Illinois River survey, and we knew that it had been defaunated by a lot of the pollution that had come down. This was before the Clean Water Act. And then there had been a little bit of a survey effort, I think in the late 80s, early 90s, and there was a little bit of recovery, not necessarily in the number of animals. There was several different species that had come in. And so this was great for all of us to be able to get in there. We blitzed it for 2 days, 20 some personnel. We found 23 species, over 14,000 mussels, and then one federal species that was thought to be extirpated from the state, the rediscovery of scale shell. Kevin actually was one that found it. And so, lots of exciting things that we were able to do on this opportunity, and lots of recruitment that we saw. So it was super exciting to see the recovery that had happened in the upper Illinois River here. So some of the other work that we've done on state wildlife grants, Allison and myself with defining expectations for mussel communities. We were doing some distribution maps, and we also helped with updates to the Streams Campaign and mussel species in greatest need of conservation for the Illinois Wildlife Action Plan that you can see here on the right-hand side. And the action plan helps provide guidance in areas of research and things like that for the state of Illinois. We also did several different modeling, random forest regressions that helped model relationships between mussel populations and their environment. And in addition, we also modeled like historical and pre-settlement distributions of about 45 of the mussel species in Illinois. And you can find all this information on our website or contact me. And some of this also is in publications. So even though I said we haven't done too much in the Illinois River, I'm going to highlight another couple of Illinois River studies. So like I mentioned before, there's been lots of biologists that's kind of studied and cataloged the mussel diversity in Illinois. And because of their efforts and then especially the efforts of Kevin Cummings, the INHS mollusk collection is one of the most premier collections in the country. And so with the combined use of museum specimens and modern day animals, Dr. Andrea Fritz had investigated mussel's age and growth rates and disturbances that happened during his lifetime in the Illinois River. And so she had encompassed like about a 140-year span and looking at the story of what these mussel shells can tell us. And so, for example, the graph in the middle is showing mussel shells that were collected in 2013 that she did for her modern animals. The results had shown that there was a 20 millimeter larger, these animals were growing 20 millimeters larger and at a 50% greater rate than mussels that were collected in 1897 from the same locations in the Illinois River near Havana. And so what else she was looking at too was studying legacy contaminants and how those concentrations had changed over time. And she looked at stable isotope signatures, which can shed light on some of the different nutrient sources in the Illinois River. And so this is an article in an INHS report, but she also published a paper on this. And then one of the other things that Andrea and Allison and Rachel Wenzel and myself did was investigate the presence of Glaucidia on Illinois River fishes, and this included our native and non-native fishes. We were interested in what impacts maybe silver and bighead carps were having on mussels, or in particular Glaucidia. And so we collected. Well, we had actually a lot of people had helped provide about 125 carp for us, and we processed their gills. We did not find any presence of Glaucidia on the bighead or silver carps, which it wasn't really that surprising. The Illinois River is at fledge stage during some of the summer of 2014, and so it could have perhaps diluted Glaucidia that might have been in the water column. We didn't find Glaucidia on any of the other native fish species that are column swimmers. We did, interestingly enough, find cell hemorrhaging and some of the carp gills. We also looked at common carp, but we didn't find any natural infestation of common carp. We also had looked at about 11 species of native fish, 290 individuals. So 11% of those had Glaucidia, and 5 species of the 11, so about 45% of those fish that were collected carried Glaucidia. So one of the other things we work on is reintroductions. We've only had primarily one main reintroduction of 2 federally endangered mussels, the northern riffle shell and club shell. There's a picture of those on the bottom left. And in their recovery plan, it's a federal recovery plan, it's in there to try to establish viable populations in 10 separate river basins throughout its range, like throughout the Midwest. And the Vermilion River in East Central Illinois was a pilot system to assess the translocation success. So this came about because there's this bridge up on the upper left-hand side was slated to be removed. And how they had to remove this particular setup was dropping it straight down the river. And fortunately, unfortunately, it's loaded with mussels right there. It's a gorgeous spot. All of us have been there surveying, and there's numerous thousands upon thousands of these mussels, a common species and a lot of listed species. And so we were able to get hundreds of riffle shell and club shell to augment into the Vermilion River. Other states did the same thing, like Indiana and Ohio. So, you know, all these places had these species that were extirpated, or river basins where these species were extirpated. And so we were translocating these animals into the system. And they were pit tagged. So this is a passive integrative transponder that we can then track. And so we're interested in both detection and survival. So there's post-monitoring of translocated animals. We did a big pre-survey work of the entire basin to help select best candidate sites and then also assess the current mussel assemblages. So this has been happening over the last almost decade, probably not quite. And then we have a grad student currently, Hugo, whose project is focused on modeling suitable habitat for surrogate mussel species in this case. So the mussel assemblage that we find here regularly in the system, and some of the mussels assemblage that's associated with like the club shell and the northern riffle shell. And so here's Hugo, and then that's actually Allison doing some of the work this summer. And then this is a picture from beforehand. So we monitor by the antenna, the hand unit shown on the previous slide. And then this is also a floating antenna that allows for kind of longer trekking because these mussels are moving. They're either getting washed out and or they may or may not like the spot we place them. So a lot of our work now, for example, I mentioned that Allison and I had done quite a bit on state wildlife grants or other grant-based work. We do a lot of work now for the IDOT and tollway contracts. And so we do pre-construction surveys. And so here is an example of Allison and her crew that are doing a pre-construction survey by transect before a bridge that's getting slated to have some sort of construction activity that would have some in-stream impact. So these mussels get glitter tagged here on the right. This species, the Theloderma metanevra monkey face, is now listed, and so it would get a pit tag. And then these animals are monitored after their movement. They're just moved, you know, not too far away outside of the construction impact zone, and ideally, you know, and a best suitable habitat or where there's other mussels that would be nearby. And so this is a little bit of a post-construction project that I've been working on for the last several years. There's a lane expansion project in I-90 up by Rockford over the Kishwaukee River. In 2013, there was an experimental translocation study with about 100 animals that estimated survival and detection until 2015. And then in 2015, I implemented a population study surrounding the portion of the river, or the bridge, excuse me. And we wanted to look at the population dynamics using this long-term capture recapture study design. And so we can help assess survival and recruitment and movement. And our focus has been on some of the rare species and the SG and C species, and then also how recolonization effort occurred after the post-construction of the bridge, because they'd had some in-stream work while they were building that lane expansion. Okay, So a couple of our newer projects is with environmental DNA. Over the last decade, environmental DNA or eDNA methodologies as a survey tool to detect invasive and then also rare freshwater species has become more commonplace. So aquatic organisms can be detected using eDNA methods, and this can often be at a higher sensitivity than with conventional methods. So what is eDNA? eDNA is genetic material that may be sloughed from an organism into aquatic ecosystems and remain in suspension. And then this can be captured in water samples. So the water samples are then filtered and processed. And you can have a species specific or a multi-species assemblage, which is called metabarcoding eDNA approach, then to detect the presence of that DNA or absence of that species. And so in this slide here, we have an example of a study that my colleague Jeremy and I participated in with a graduate student. You can see Amanda on the upper right. She'd had a few different eDNA projects. This one, we were looking at the role of stream flow on eDNA concentrations, and then the detectability of corbicula. So corbicula was pretty much our study animal. They are present. We had two rivers that we looked at in East Central Illinois. They were present and abundant in both of these. And so we wanted to see over the course of a year, how does eDNA change given temperature and stream flow and things. And so the results of this study was that we found that higher stream flows actually decreased eDNA concentrations. Flooding can produce false negatives or non-detections where the actual animal is. And so we determined that the best time to collect is in low flows and base flows, and also when your species of interest is would likely have the most eDNA, it's sloughing the most DNA into the system, which would be like during reproductive events. And so a second study that I've recently pursued is with native mussel species and its host. We have the salamander mussel on the upper right. Since we have no pictures of really good live ones here, I borrowed one from the nearby state. And presently, salamander mussels are listed as endangered in Illinois. They're species of concern or threatened and endangered in most surrounding states. And then mud puppies also are listed as threatened in Illinois and a species of special concern in several surrounding states. And so determining the presence of salamander mussels is difficult using the conventional time search methods because they're small, they're rare, and they actually like to cluster under large slab or flat rocks. And Clark in 1985 had published that finding live salamander mussels are a most unusual occurrence. And he'd also went on to report that Baker in 1928 and Frierson in 1927 also, reported not finding hardly any live specimens. And so there's been one targeted survey effort in Illinois, but that was in 2002, and no live individuals were detected. There were some recent dead or weathered shell. And so given the complexities of finding live salamander mussels with any regularity, I wanted to try this proof of concept study using eDNA as a survey tool to detect their presence or absence. And so also wanted to do that with the mud puppy at the same time, because mud puppies also shelter under rocks or trees and things like this. So we had a known positive water collection. This is Genoa National Fish Hatchery are actually propagating salamander mussels and augmenting them back into some of their streams in Wisconsin. Here's an example on the lower right-hand side of the filtering process after we've collected our samples. And then an example of the stream. This is the Emberell River, that one of the sites that we've been working in. And so lastly, real quickly, Jeremy and I have been pursuing this question of corbicula. Excuse me. Because we discovered in 2015 a new Corbicula in Illinois, we were actually searching for more scale shell. That was, we'd found in, Kevin had found in 2013. And so there had been a little bit of an effort for a couple of years to see if we could find any more. But we actually found a new invasive species instead. And so we started to pursue this question about, well, where are they? And so we had an intern from the NGREC program in 2016, I believe, where we did some extra survey work in upper Illinois River. And then we've been collecting records from other folks that sent us. You can see here now in the Midwest of where we've seen Form D pop up. And so something to keep an eye out as you folks are headed out into your rivers to see if you have any of these pink little stripy ones showing up. The Form B, the Largotilleri, had showed up in Illinois in 2008. And then Form A, or Corbicula fluminea, has been around since the 50s, 60s. And we also have some interest in documenting where zebra and quagga mussels are, the Corbicula species that I just mentioned, and then a couple different mystery snails that are popping up in the state as well. So with that, I'm going to go ahead and wrap up, and then I'd love to take some questions. So here we are, the INHS Mollusk Lab. Jeremy and Allison, Kevin and Rachel, we've had a variety of projects that we either work independently on or together on. And then there's some resources on the right-hand side where you can find some great information for mussels. And with that, thank you for your participation. And I'm gonna, happy to figure out how to take questions. How do I open the chat box?
All right, thank you very much, Sarah. That's a great presentation, a lot of wonderful information. The chat is filling up with questions, so just so you don't have to try to read those, I will go ahead and read those for you as they move up my screen. I will try to read them. All right, so first we have a question from Newbie Thomas. Ocean acidification caused by the CO2 that humans put into the atmosphere results in shell deformity in calcifying organisms. Does this affect freshwater mussels too? And if so, how bad is it in Illinois?
That's a great question. I don't think that anyone's really looked too much at that. There has been a little bit of research, actually. One of my colleagues here at INHS, Dave Suchek, has looked at road salts around Chicago region and how that's affecting some of the local streams and invertebrates in those streams. And I'm trying to recall, at least for some of the animals that he had tested, I know a couple of the mollusk species that he had tested, there wasn't too much impact, if I recall. Could be wrong. I haven't looked at that in quite a while. And as far as shell deformities, though, I don't really know how to answer that. I'd have to try to look that up. There's been a little bit of information on shell deformities, but I'm not sure about that as far as how salt would affect that.
All right, then Phil Nicodemus is asking or saying, oh, says mystery snails sounds like a great name for a football team. So like, sorry, I thought that one was a question. All right, so Robert Cantor is asking, can you say in general terms whether mussels in the Midwest are doing better or worse now than they were about 20 years ago?
Thanks, Rob. That's a great question. 20 years ago, I don't think I'll speak to the entire Midwest. I think I'll keep it to Illinois because in some places, they're doing fine, and in other places, so real quickly, actually, we're going to participate in a regional study on enigmatic mussel declines. So several different places in the Midwest have had these enigmatic mussel declines. And while Illinois has had significant decline initially, we had seen some really great rebounding of mussels in our streams. I think only really within the last probably 15 years, we start to see a little bit, so the Clean Water Act was in the early 70s. And then it still takes decades for mussels to recover, just given the longevity of their lives and how they recruit and things like that. So in some places, they're doing better than what they were 20 years ago. And in other places, they're doing worse. I'm sorry, I'm not really answering that question very well.
Sounds like the answer is it depends.
Yeah. I'll just stop talking. I was like, I'm not.
Okay. Angie offers that if you are looking for more information on pearl buttons, you can go to muscatinehistory.org, which is the National Pearl Button Museum.
Okay. Great.
John Griffiths is asking, do Glochidia adversely affect the health of their hosts?
That's a great question. No, they do not adversely. Like, while they're technically considered a parasite, they do not adversely affect their host. Now, and the only cases that we do know is like in propagation, if, for example, they get over inoculated, it's called, when you inoculate a host fish, you're exposing them to the Glochidia to attach and stuff. So if they, for some reason, get over inoculated, so there's too many Glochidia that end up on the gills, then yes, they can die. That was discovered pretty early on, and actions, appropriately figured out from then. But no, most of the time we don't really see that, especially wild caught fish. There's usually not really that adverse effect.
Okay. And then we have someone asking, where can I find Kevin Cummings' Mussels of Illinois book?
Yeah, so we've had a lot of changes on our website, but you should be able to find on probably the publications of either the Prairie Research Institute. You can find a link to the Illinois Natural History Survey, or either that or the Institute has a publication page. And I think you could still find it there. Or you can contact Kevin himself and maybe he can send you a copy. I'm not sure if he has copies or not, but I will give you a heads up that there is a new field guide in works. I don't know when that'll be out, but I know that that's been in the works the last couple of years.
Okay. And just to kind of interject kind of from the bottom here, regarding newbie's question, the Great Lakes and the surrounding region are on limestone, which is a basic basic in nature, and so acidification is mostly buffered in this region.
Thank you.
Yeah. All right. And Bill Bromer is asking if we have any suggestions on how to verify mussel identifications. He has taken the RiverWatch mussel training, but is not confident in his ability to identify.
Yeah. Lots of practice of handling shells, getting out there. You can take pictures. If there's a teaching collection, like if you're like a RiverWatch coordinator, different things like that, as far as finding a teaching collection. And yeah, just getting out there and seeing how diverse they are. Some of them are very difficult. I mean, some species look very similar. And then even the same species in different basins can be slightly different, that you just do a double take. So a lot of that's just learned, and just give yourself time to keep practicing.
Okay. What's the best way to explain to the public the importance of native mussels filtering water when people typically only hear about mussels being a bad thing, like over filtering the water in the Great Lakes?
Sure. I think pointing to a little bit of those ecosystem services that I had touched on, and that example of just what they filter, for one, and then how that contributes to human health ultimately. I think would be a couple of things to think about. The other thing that comes to mind as far as than just like being able to celebrate the biodiversity of native mussels and their uniqueness, as far as their intrinsic value, I think to here. So, those are a couple of things. I guess I would come to mind immediately.
Okay. Let's see. Jean asks or says that you mentioned that mussels are being used or considered to being used for cleaning up aquatic environments and that contaminants are either excreted or stored in the tissues. What happens to the contaminants after they've been filtered through the mussels? Are the mussels that absorb the contaminants in their tissues then considered contaminated, and how is that handled?
Yeah, that's a great question. I don't fully know the answer to that. I know some of that can get stored into their shells. And so that's essentially like sequestered. Even though shells could stick around for a long time, you have that's then sequestered as opposed to, moving out into the system. And so, and it's and similar concept with that being in the tissues. So the problem though that you can run into is that bioaccumulation effect, right? Well, if something eats the mussel like a fish, well, then how much of that's going to actually bioaccumulate in the fish, and then so on and so forth up the chain. So I think that's still a good question to think about, particularly in some of these places where there's like very heavy metals and some of these other types of chemical components and stuff. So as far as the amount that's in the system that's not being diluted, I think would have that significant effect on the mussels in there. So yeah, it's a little bit of a catch-22, right? But that's definitely something worth thinking about.
Okay. Let's see. Aubrey asks, do you recommend tagging live mussels for general citizen science population projects?
Yeah, that's a great question. I think yes. I think it could be very interesting. So we don't have regular surveys in Illinois outside of like at consistent places outside of RiverWatch effort now. The survey effort that we did was this one time on a grant. It's not a rotating survey. And so being able to tag these animals that you are going back and surveying every year at a site, or every other year, I should say, depending on how your sampling strategy is, I think that could be very informative. And we see a lot more of this great citizen science information that is also being incorporated into other research and stuff as well. And so, especially in places where we haven't had much surveys in Illinois. And so being able to generate some of this kind of long-term data is beneficial. But, the caveat is not stressing the mussels out too much. And there's ways to, for those sampling techniques and stuff like that to help with that.
Okay. Yeah, and I'll just add on to that. If this is a project that you're interested in doing, I would definitely recommend reaching out to Sarah off this later on to make sure that the method you're using for tagging is going to work for mussels, and to make sure that, you can get any useful hints and tricks.
Yeah.
All right. David asks, is there a timeline or order for when species came back after the last glaciers?
Man, all these great questions. Kevin Cummings would probably be able to better answer that than me, or Dan Graff. They've had a lot of kind of that larger scale look. Yes, I think so. And I'm not brushed up enough on that to really answer that question very well.
All right. Jake asks, do any freshwater mussels in Illinois consume blue-green algae, or are they affected by harmful algal blooms?
This is something that I would love to see some more interest and research happening. So, I mean, really within the last 5 years, right, there's kind of been this explosion of research and thinking about harmful algal blooms. And so we haven't seen a whole lot of that information yet in the mussel literature yet, but I expect that to be coming. And so I don't really know the answer to that, but I think it's very much worth investigating.
Is it something that they are filtering though when they pull in like the water from the food though? Like for food?
Yeah, I'm sure that. Yeah, well, so I guess, hold on. So I guess, one of the things that clams have the advantage on is being able to close their valves, right? So I don't know how they could determine, or if they're determining something that's a toxin, bacterial toxin, and how long, like if there's this big bloom that happens, part of our thought is, well, they just kind of clam up until it passes, or how they sense that, I have no idea. And if that's the case, I don't know yet either. So there's maybe a little bit twofold to that question. That yes, maybe some mussel species might, while others probably don't. And I think that depends on if it's getting too hot. Some mussels will just bury and hunker down and wait it out, if that's part of the trigger for some of these blooms that happen. And then other mussel species are moving and trying to find better temperature, better areas. And so if there's still that activeness that's happening and during the bloom, then they'll be taking in that bacteria.
Let's see. Aileen is interested in the use of mussel shell as temper for pottery during the Mississippian period. Are there any species currently in Illinois that would have been plentiful and may have been used on a massive scale 1,000 years ago?
Yes, yeah. I think so. A lot of the thicker shelled species are very long lived, and we know that a lot of these thicker shelled and big river species today were present a long time ago. And so there's been some really interesting, in Wendell Hagg's book, he has a little bit more of the information on some of this prehistoric use, and just the amount of shells and middens. And so yeah, there would be some of these species. I think probably some of your thicker shelled species, the stuff, and then there's even another species that was used very much sought after for buttons that I was reading about, which is the yellow sand shell. What else was the other part of the question?
I think that probably covered it. Okay. And then Newbie has a follow-up. Looking at Dr. Fitz's research showing mussels are bigger compared to 1897, does this maybe result from rivers receiving nutrient pollution from agricultural fertilizer, or do mussels somehow simply have better nutrition now than they did at the time?
Yeah, so I think part of that theory was whatever's coming from Chicago, along with what's coming in from the ag, has just provided that extra food source. Yeah.
So, and kind of my own follow-up. So it's more of a food source issue rather than maybe them being eaten more readily by certain species, and just evolutionarily they have to adapt to grow larger faster so they don't get eaten. Hypothesis.
Yeah, good thoughts. One of the species that she looked at was the tree ridge, I believe, and that's a thicker shelled species, and so I don't think that kind of pressure would be on that species. I think more so perhaps on some of the thin shelled species, which is what we see more dominant in our systems now. But we still have like the tree ridge that I think was one of her main animals of study in her research. And so I think it's just probably the former, as far as the water quality improved, but in addition to the water quality improving, so them to stay alive long enough, is that there's just all of these these added nutrients now in that improved water quality. And so that's probably contributed to some of that faster growth.
Okay. And have there been any efforts, to your knowledge, to help reintroduce mussel populations into more urbanized waterways?
Yes, actually, thank you very much for that question. Actually, the Urban Stream Research Center in DuPage County in Chicago is doing this. They're doing some fantastic work with common mussel species in the DuPage River. And so like DuPage was hit significantly and had a lot of loss of not only species, but just the abundance of mussels. And so, so Jessie DeMartini and her crew at the Urban Stream Research Center there at DuPage County Forest Preserve is propagating some of the common species and being able to put back into the DuPage and the West Branch DuPage had a lot of restoration work over the last several years as well. And then she's also has a couple of other target species that she's working with in some of those streams in Chicago region.
Okay. And then I also had a question before I get to the last ones in there. So you mentioned and showed some pictures of the different types of corbicula of the Asian clam or Asian mussel. And so from what I could tell, the visual difference between them was easiest to see on the inside as far as the different coloration. Is that something like, I guess, first off, how long after they die is the color prominent enough that you would be able to use that as a way to tell them apart? And then also, if a RiverWatch volunteer sees different color, multiple colors perhaps, or the more pinkish color of the new variety, should they take a picture and send it to you or to me to send to you?
Mm-hmm. Yeah, no, that'd be fine. Definitely. And it should like that, the shell coloration should excuse me, remain, I think, pretty well for, I think probably depending on stream system, maybe a year or two, maybe more. Some streams are a lot harder if it's buried, but especially if it's fresh dead that season, it should be pretty easily seen in those color differentiations. And then if you do come across the shells then of any of those new corbicula, then you can take a picture of them, send them to me. That'd be totally fine. Or actually, we would love to have those vouchers as well. So you could just save the shell, or you could ship them to me, or figure out a relay on how to get shells. So, or, I'm going to come out and try to do a site determination and stuff. So, especially with any of the smaller tributaries in Illinois, right now, that species is in the Illinois River, and we've seen them in the lower part of the Des Plaines, but I haven't seen the form D show up in any of the small tributaries. We've seen some of the B, the purple form, show up in some of the tributaries though. So.
Okay. Jake asked, do urbanized areas with more frequently occurring flash floods affect the ability of some freshwater mussels to exist in a stream or river?
Yeah, definitely. They can either be dislodged in some of these really intense flooding events, and they can be transported to other areas that's not as suitable habitat, so they could die. There's depending on the intensity of floods, I didn't talk about it in this talk, but one of the other projects that Alice and I worked on with Sarah Andre is looking at some restoration criteria of freshwater mussels. And so we looked at two species, the ellipse and spike, and those species had differing factors that limited their ability to survive. But one of the factors that helped, I think it was spike, was this different degree of flood events and stuff. And so different species kind of require some different flow patterns and things. So yes, it can have a lot of times, especially some of these flash flooding, I think can have some negative effects on mussels, and primarily dislodging them out of where they were existing.
Okay. And then from Megan, will mussels be able to adapt or range shift to mitigate against climate change impacts, or would humans need to intervene and relocate the mussels and perhaps even their host fish?
Yeah, that's a great question. There's, we see some of that with some of the range adaptation, some of the range shifting that we've seen, expansions of some of the mussels, a little bit of what I've highlighted, but then some others. And so some of these species that that we see that's pretty dominant now, and especially in our large rivers, are the species that are on fishes like freshwater drum that I don't know if they're migratory per se, but they run up rivers a lot, and then they are fast growers. They have a very effective strategy and plenty of hosts available. And so these mussels with this kind of strategy of being able to mature quicker, grow faster than perhaps their counterparts that are like the thicker shelled, longer lived animals. So those are the ones that we're going to see adapt probably better to climate change impacts than some of these other more sensitive species or some of the other rare mussels that are still rare would probably have a more significant effect of climate change.
Okay. And then Jake is asking when we'll have another mussel identification course for RiverWatch. So that one, I guess, is for me. The earliest we would probably be doing something like that would be the fall. I'll certainly keep that in mind, that we have people who are interested, especially after today with the great turnout at this Zoom RiverWatch
Zoominar.
So yeah, we'll definitely have to look into that, and maybe Sarah can help us out with putting one together.
Yeah. I like taking people out to the river too, so happy to help with that.
Great. Okay. Well, I think that's all the questions. We actually made it through them all. Sarah, I want to thank you for the great presentation. There were several comments of thank you and great job but I skipped those in the questions. And thank everyone for coming tonight. Before you go, I did want to mention a couple of upcoming events that we have. Our next winter speaker series will be next Wednesday, February 17th at 7 p.m. And Katie Whitlow will be talking about underwater scuba adventures in Illinois. And then also next week, we're very excited to have a one-of-a-kind Zoom event, an evening with Dion Thomas, the voice of Illini basketball. Tickets for this chance to meet an Illinois basketball legend are $25, and all of the proceeds from that will support RiverWatch. You can learn more about these events and other future activities on our webpage or on Facebook. And again, thank you all for coming, and I hope to see you again soon.